Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Menarini Silicon Biosystems, I'd like to welcome you to Dissecting Genetic Heterogeneity in Brain Tumors using Digital Sorting of Single Cells. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. I'd like to introduce our moderator for today. He is Dr. Niccolo Manaresi, co-founder of Menarini Silicon Biosystems. Welcome.
Thank you, Elizabeth, for the introduction. I will be moderating the webinar today, which is about dissecting genetic heterogeneity in brain tumors using digital sorting of single cells. And we have two speakers today. We will have first an introduction from Dr. Raimo Tanzi, my colleague, Chief Commercial Officer at Menarini Silicon Biosystems, which will provide an overview of the DEPArray technology and a short introduction of workflows which we support for tissue analysis and in particular for FFPE. And our second speaker will be Matthias Nudel. He's a medical director and a medical director of molecular pathology and diagnostics at the Department of Pathology and New York University Langone Medical Center and Medical School. And he has been pioneering the DEPArray application in brain tumors and will share with us some very interesting, also unpublished data highlighting how DEPArray helped him in addressing the challenges of disentangling the heterogeneity of brain tumors with an emphasis on epigenetic analysis. And with this, I hand over to Raimo for his presentation.
Okay, thank you, Niccolo. Thanks, everybody, for joining this webinar. I will go through a brief introduction of the technology that is at the base of the work which is going to be presented in the second part by Dr. Snudel. The technology talking about is called the DEPArray, and the DEPArray stands for de-electrophoretic array. The system is composed by a benchtop analyzer and by a disposable cartridge device. The device is the place where the cell sorting happens, and this device is the first example of a joint microfluidics and microelectronic device. And the sorting of the cells happens, is run in this little window that you see at the very center of the cartridge in this picture. How does the technology work? We can see it in this animation showing a section of the flow cell. So here you see the ceiling, which is a transparent glass, which helps us see through it what's going on inside the flow cell. And the floor. The floor is the active part of the flow cell and is paved with several hundreds of thousands of microelectrodes, each of which is controlled by software through the semiconductor CMOS chip, which is at the base of the cartridge. So we can position every single electrode in a positive or negative charge as we like, building any kind of geometry we want. And in particular, when we generate a geometry like this with a square of 3 by 3 electrodes positioned with the outer 8 in a positive charge and the inner one in a negative charge, we create on the vertical projection of this square a situation that we call a de-electrophoretic cage. Basically, what we generate by doing this is a point of minimum energy, which is standing right on top of the negatively charged electrode. By doing so, any object which is floating in the flow cell, the moment we activate the de-electrophoretic cages, has no other option but to go and occupy the position of minimum energy. And when the objects are sitting in these positions, they cannot move, and we can observe them through the ceiling of the flow cell with the microscope that is embedded in the system, and we can make the selection of the desired objects. And once we know which cell or cells are the ones that we are interested in this experiment, all we have to do is tell software where they are located, and software knows which pixel, which electrode is controlling that cage, and by moving the negative charge one position at a time on the floor of the flow cell, it can accompany the cell out of the flow cell into a collection chamber and ultimately in our collection tube. The detection, the selection of the cells is done with the help of a microscope, which is equipped with fluorescent channels, so we can use immunohistochemistry markers. We can use DAPI to select nucleated cells, but also to verify the ploidy of the cells that we are selecting. But we can also use images, so we can see the size and shape of the cells on each one of the cages, and so combining all this information, we can make our selection of the desired cell, and then the device is going to take it home for us. So the principle and the unicity and the innovation of this technology stands in the fact that we leverage on the extreme power of CMOS chip technology in controlling pixels. We know that this technology has 100% precision in controlling pixels, and the invention, or the let's say, the innovation stands in using these dielectrophoretic cages to bind objects to the pixels. This way, we are transferring the precision of CMOS chip in managing pixel into a precision of DEPArray in managing objects. And on top of the whole system, we have this microscope scanning, which is basically the referee, which is helping us determine whether a cage is zero, so is a cage that either is empty or contains unwanted cells or contains too many cells, or a cell is a 1, that is a cage that contains only one cell and of the desired kind. So altogether, this is the first example of what can be defined a digital sorter of the cells. Just one more look into the operation of the device. The flow cell that I showed you in section before, here is shown in a top view, and you see that it's organized in 3 chambers. The main chamber is the largest space of the flow cell. Here we can generate up to 30,000 depth cages, so 30,000 parking places, let's say, for the cells. So here is where we load the stained suspension of cells. Cells are going to occupy randomly the cages available on the chamber. Once we have identified, for example, that these and these are the 2 cells that we are interested in our experiment, software is first going to find the fastest route to move them into what we call a parking zone. And from the parking zone, the operator has 2 options. One is if you're interested in single cell analysis, you can have software to move the cells one by one into the last chamber, which is called recovery chamber. And here you finish the digital control of the object, and a very analog droplet of buffer is going to wash the cell out into your recovery tube. The second option of using this sorter is not for single cells, but for just pure cells. So supposing that instead of one cell in the parking, you have like 200 cells of the same kind, and you are interested in collecting all them together, software is going to move them all together in the recovery chamber, and then all together they will be deposited in your collection tube. So this is both a single cell device and a pure cell device, depending on the way you want to use it for your research. What do we do with this technology in oncology? Well, we work in many areas, but the 3 major areas of interest are liquid biopsies, where we are enabling the collection and isolation of pure single circulating tumor cells for consequent genomic analysis. In the solid biopsy area, we transform an heterogeneous FFPE tumor sample into a collection of pure tumor cells and pure stromal cells, so that you can proceed with a genetic analysis of the 2 components separately, and which I will show you a couple of examples of how this can improve the quality and the accuracy of the analysis. And lastly, in the area of immunotherapy, this time working on fresh tissues and not on fixed tissues, we can disaggregate the cells, and on top of selecting tumor and stromal cells, we can select tumor-infiltrating lymphocyte cells, which can be collected live and can be even cultivated after the recovery, or can simply be used for transcriptome analysis as desired. Today we are focusing mostly on the middle application, the analysis of solid biopsies. So I just want to give you a couple of views on how we process the sample and what kind of improvements you can obtain in the genetic analysis of these kind of samples. So we start from a paraffin block from which we can either cut a slice or we can punch. And whichever way we proceed, the sample is deparaffinized like a normal FFP procedure, and then the tissue is disaggregated enzymatically, stained with antibodies that recognize the tumor cells and stromal cells or other cells, as you will see today in the presentation from Dr. Schnuderl. And then the DEPArray is going to detect the two groups of cells, and they're going to deliver a tube with pure stromal cells, a tube with pure tumor cells, and we can then proceed with their analysis. So far, we've been developing this application for a couple of years, and so far we have demonstrated that these cells are amenable for FISH immunohistochemistry. You can run NGS cancer panel based on amplicons. You can also make a genome library for NGS analysis of whole exome, whole genome, low-pass genome. And today you're going to hear how Dr. Schnuderl has developed his workflow for methylation analysis. You can clearly realize how all of these methods benefit a lot of the purity of cells because what you find in a pure cell population, you know, comes from that population. There's no doubt that is coming from the other one and is not hidden by the other contaminating population. So to show you some concrete things, this is the starting sample, the way you are probably used to see it, on the pathology slide with a mixed presence of stromal and tumor cells. And this is the way we see the same sample after we have processed it on DEPArray and image it on the software through a scatter plot based on the fluorescence intensity of vimentin dye and cytokeratin dye. As you see, the cells here form four quadrants. The first quadrant is that defining the stromal cells. This is defining the tumor cells. Here in the middle, you see a large number of objects, the vast majority of which are heterogeneous cell clusters made by two or more cells that didn't separate during the disaggregation process, because since one is tumor cell and the other one is stromal cells, they carry both fluorescent dyes, and therefore they are a double positive objects in the middle of this chart. But if you do a careful inspection of all these objects, you will be able to find, rarely, but you will find single cells, which we can clearly define as single because we have the image of each one of these objects, and these are cells that are genuinely single, genuinely double labeled, and therefore they are a different kind of cells from these and from the tumor cells, and they are cells that we can probably define as epithelial mesenchymal transition cells due to the fact that they express both markers. Another important parameter that we obtain during the sorting of cells through the DEPArray is the exact ploidy of the cell groups. Diploid level is indicated by the peak of DAP intensity obtained through the analysis of the stromal population. And the analysis of the tumor cell population is giving us a different level of ploidy. The ratio of these two peaks is basically what we call a DNA index, and is a measurement of the exact ploidy of the tumor cell populations, which is a very useful parameter you will see for a normalization of the copy number aberration data, which is going to enable us to achieve very precisely an absolute copy number aberration profile. To give you an idea of how different is the analysis of pure cells versus mixed cells in a tumor, I'll show you a couple of examples only. The first is a whole exome analysis. This is a pancreatic tumor, which had a pretty low cellularity, between 10 and 15% was the estimate from the pathologist. The first slide shows you the variants that were found on the unsorted sample, and you see there are 4 variants at different variant frequencies. All of these variants, by the way, are irrelevant for the, let's say, gene. They're just SNPs, and this is all that could be found at the level of the p53 gene through this whole exome analysis. When we analyzed from the same sample the 2 separate populations, tumor and stromal separately, the first thing that we could find was a very evident somatic variant, which appears in 100% of the DNA molecules coming from the tumor selected cells and present in 0% of the DNA molecules coming from the stromal selected cells. This is a variant that was actually present in the unsorted sample, but unfortunately the variant caller couldn't call it because it was only represented by 1 read out of a total coverage of 20 at that position, and there's clearly no way you can call a variant with a single read only. But when you separate the 2 populations, it's absolutely unequivocal, the detection of the variant in the tumor. And these numbers are also giving us a very interesting and clear view of the precision of our digital sorter, because seeing 100% here and 0% here is a clear let's say, example that tells us that there's no stromal cell accidentally present in the tumor population and vice versa, because otherwise these numbers wouldn't be a 0 and wouldn't be 100. But on top of being more sensitive in seeing and detecting somatic variants, we can also see other things by analyzing the pure cell populations, because when we see the complete picture of this p53 gene, we see that, first of all, the 4 variants that were detectable in the unsorted sample are all germline variants, which are nicely detected at an almost precise 50-50 ratio as expectable in the stromal cells. But we also see that when we move to the tumor, the variant allele or the wild type allele is lost by the tumor cell population. So this helps us reconstruct that the tumor, this patient was born with these 2 haplotypes at the level of his p53 gene, that the red haplotype was hit by the somatic mutation and the blue haplotype was lost due to a loss of heterozygosity event. So this is a very clear, detectable dual hit event at the level of the p53 gene, which unequivocally tells us that this gene is completely lost by the tumor of this patient. Another analysis that benefits a lot of the purity of cells selected by DEPArray is copy number aberration. This is a plot generated by low-pass genome analysis by NGS of the pure stromal cells, nicely flat, nicely clean, as one would expect from a normal genome. This is the plot obtained from the unsorted cells. This was a 40% tumor cellularity, so not super good, but not even the worst kind of sample one can find. And clearly this is aberrant, so we can surely say that this is or contains some tumor. But the moment we want to stratify this patient for some features like long-term survival or respond to a drug or something, we have a few grip here, because everything is very much concentrated around the noise level. There is only 1 spike here and maybe 1 spike here. Losses are very difficult to detect. But when we purify this, so when we remove the stromal contamination and we analyze the tumor in impurity, this is how the profile looks like. So we are finally exploding, let's say, the informativity and the information that is contained, is written in the genome of this tumor, and we make it readable, pretty much like if we were using a fuzzy scanner for reading barcodes in a supermarket, and we are changing to, let's say, sharp barcode reader, which is giving us the power to use the full potential of these information that is coded in the barcode in this example and coded in the DNA in the example of the tumor. And once we have done this, and once we have normalized the copy number using the DAPI ploidy that is determined directly by the DEPArray, we obtain, and we can do a comparison like this. On the bottom chart, you see the B allele plot generated from the exome data from the sample, where you see there are some balanced alleles regions, there are some imbalance regions, and on top you can see the absolute ploidy plot of the entire genome of these patients. So we can now go and dissect the information that is present in the genome of this tumor. Here we see, for example, chromosome 3 as an imbalance, which looks like 66-33%. And in fact, when we go to the copy number, we see that the entire chromosome has 3 copies, which correspond exactly to a gain of the red allele versus the blue allele. Here you see another example on the distal part of chromosome 6, where the B allele plot tells us there is clearly a loss of heterozygosity because alleles are only at 100% or 0%. But if you go to the copy number, you see that the number now is 2. So if the number is 2, we can assess that this was the initial genome, the blue allele was lost. But instead of having just 1 copy of the red, we count 2 copies. So there's been an amplification, a further amplification of the red. And even more complicated is this situation where you have an imbalance, but the number we see is 5. So we can tell that the copy number has gone from 1 and 1 to 2 and 3 for the 3 chromosomes. And the last thing that purity enhances and enables to do very clearly is the analysis of a simple cancer panel. So if you want to investigate on what relevant mutations, cancer mutations are present in a patient, you just run a cancer panel. And in this example, you can detect 3 different kinds of genomic variations. One is these 3 events where you see the stromal cells show 0% of these variants, and they move to 100% in the tumor cells. This is a sign of a dual hit event, like we saw before on the exome data. This means there is a SNP and there is a loss of heterozygosity. If you see something moving from like 50-50 in these 2 cases to 100% or 0%, this is a sign of just a single event, the loss of heterozygosity. And if you see something like this moving from 0% to 50%, then you are in front of a simple heterozygote somatic mutation, but still the other allele is present. All this information is clearly much more difficult to extrapolate when you work on the unsorted, and this is the kind of data that you can obtain, which is basically barely telling you a yes or a no, there is a mutation or there is not a mutation in the sample. With this, I close my part and I just leave the microphone to Dr. Schnudel.
Thank you, Raimo, and let's welcome Matthias Schnudel.
Hi, everybody. Thank you all for joining us today. So we'll be talking, after this nice introduction, about DEPArray system. We'll be discussing specific applications of DEPArray that we have developed here at NYU with the help of Silicon Biosystem to dissect the genetic and also epigenetic heterogeneity in brain tumors using this unique platform. So just a little bit of introduction. Why do we need to study tumor heterogeneity? Because I assume that not everybody in the audience deals with primary brain tumors. So malignant brain tumors are not the most common cancers, but they account for about 80,000 new malignancies every year in the United States. We have about 26,000 primary malignant brain tumors diagnosed every year. They have a relatively high mortality, and if you look at this part of the slide here, you can see that mortality increases, and that is largely due to gliomas, which are the most primary brain tumors in humans, And it's largely due to glioblastoma, which is the most common of the gliomas, is the most common primary malignant brain tumor, and it has a peak incidence between 45 and 75 years of age, has extremely low median survival of 1 year, and it can arise de novo from lower grade lesions. They're very repetitive genetic alterations that we are aware of from the TCGA and other studies, such as amplifications of receptor tyrosine kinases, p53 mutations, CDKN2A deletions. What is the most important is that in comparison with other cancers, there have been absolutely no success, or I would say extremely limited success, with using targeted therapy in malignant gliomas, and that is partially due to heterogeneity. Glioblastoma is extremely diverse tumor with a very important role of tumor microenvironment. It's characterized by hypercellularity, atypia, and high mitotic activity. There is an abundant microvascular proliferation and necrosis. On the genetic field, the landscape studies have shown that you can classify glioblastoma into multiple genetic subgroups that are driven by drivers such as K27, histone H3 K27 mutation, IDH mutation, and then into multiple other groups that used to be termed classic mesenchymal proneural. Now we distinguish them more as RTK1, RTK2, and mesenchymal. However, these large subgroups do not account for heterogeneity. The development of malignant gliomas is characterized by additional mutations that arise during the time. You can see WHO grade 2 astrocytomas are driven by IDH1-2 mutations and ATRX loss. And then in glioblastomas, we see amplifications of receptor tyrosine kinases, which were originally thought to be almost mutually exclusive, but I will show you that that is indeed not true. It is this heterogeneity that is largely responsible for resistance to therapies and recurrence and death. We have shown a few years ago that receptor tyrosine kinases are indeed not mutually exclusive, and they can actually co-occur in the same cancer. And there's two interesting types of intratumoral heterogeneity. You can see in this tumor where amplifications, this is FISH, where amplifications of PDGFR alpha gene in red and MET gene in green are seen in the same cancer cell. And in another tumor, these amplifications are present in mutually exclusive subclones, such as EGFR and MET. And what is important to note is this is the TCGA data here. We also confirm that these amplifications can co-occur. But the problem is that when you have amplifications in the same cell or in different subclones, you can never distinguish it by using any kind of whole genome approaches. This is just from the times of array-CGH, which we've shown is that first of all, these studies may not be sensitive enough to detect like a small subclones present in the tumor, but you also cannot distinguish whether the same gene is the genes are amplified in the same cells or in different cells. So we have gone through extensive work and show that indeed these subclones actually originate from the same precursors. This is an example of a tumor that shares homozygous deletion of CDKN2A, but tumor cells are amplified for EGFR, MET, and PDGFR alpha. We have also shown that these subclones can share the same p53 mutation. So they arise from the same precursor, but then later on in development, they develop these later drivers that can be mutually exclusive. However, the biggest question to us was, does it even matter, or are some of these subclones and endpoints of evolution and don't really matter? But we've shown that all of these subclones proliferate. They have approximately the same rates of proliferation, and most importantly, they produce protein. So you can see here MET amplified cell produces MET protein. However, EGFR amplified cells do not express this protein. So we assume that they're functional and they proliferate and play some role in development of the tumor. Deeper you go into heterogeneity, more changes you observe. And keep in mind, we are here just at the level of copy number changes. We are not even diving into mutations. Here you see an example of a tumor that has up to 4 distinct subclones. So it has an EGFR amplified cells, has PDGFR alpha amplified cells, has MET amplified cells, and then it has cells that are also tumor cells because they have a loss of P16. But they do not have any amplification. So you can be dealing with 4, and I'll show you even more subclones present in the same tumor in a relatively small amount of tissue. The most challenging part is that the number of cells, so the proportion of these subclones, can vary immensely. And that is a huge problem because you will not be able to pick it up on a whole genome or whole exome approaches, and you will probably not have enough cells to pick them by flow cytometry either. So you can see in some tumors, you have clones that are relatively sizable, but in other tumors, the amplified clones can be extremely small, and you would never pick it up on arrays or on a whole genome sequencing. So how do you get to study these subclones and see what do they do and how do they differ from other cells? So heterogeneity has been a huge topic in cancer, including brain cancer. And this is some beautiful work that was done by a group at MGH, Mario Suva and Brad Bernstein. And of course, we are also doing a lot of single-cell RNA-seq. And it's a really beautiful method. You use a flow sorting. You sort your cells. You do single-cell RNA-seq. You have a huge amount of data. And it is really a beautiful way how to deal with heterogeneity and look deeper into heterogeneity. The problem is that vast majority of single-cell profiling techniques are all optimized for fresh cells with a very high quality of DNA or RNA, and they rely on a high number of fresh cells, which cannot be really curated. You can't really handpick the cells of interest to you. But when you look at where the tissues are, the vast majority of pathology samples are archived in formalin-fixed, paraffin-embedded tissues, and they're not amenable to these techniques. And many of the molecular techniques that are used for dissecting heterogeneity on a single cell level are not optimized for FFPE tissues. So how do we go from there? So this is just showing you here that it's sort of a classic pathology block where the tissue is embedded in this wax, and then this H&E stain that helps you to look at the tumors. So DEPArray applications for brain tumor heterogeneity are numerous, and Raimo has shown you nicely how you can really dissect tumor cells, stromal cells. You can look at the mutational events, and you can really see what is actually happening in terms of genetics. I will show you in 3 chapters how we work with the DEPArray, and where we think that DEPArray can be really uniquely positioned to help you answer questions that you have specific for heterogeneity. We'll be talking about genetic heterogeneity and how to look into the subclones, how to isolate rare infiltrating cells from low-grade tumors, and then look into heterogeneity of a brain tumor microenvironment. So the workflow that we are using is a little bit different than what Raimo was presenting. We actually do whole genome or targeted profiling, either for copy numbers and mutations, upfront to identify targets of interest that are in the tumor for us. Then we create a cell suspension from FFPE tissue. We use immunofluorescent markers with antibodies of interest to us. We inspect the suspension for the quantity and quality. The good thing is that you really don't need a lot of cells to get a pure population. And then we inspect the quality of the staining. Then we perform our DEPArray runs, select subclones, handpick the cells we want to work with, isolate them, and perform downstream molecular analysis. To identify the subclones of interest, I will show you a few different ways how you could do it. We are looking with markers looking for overexpression of proteins. Then we use mutation-specific antibodies to isolate not only cancer cells, but cancer cells with specific mutations, and then markers for the tumor-associated stromal cells. We do also a lot of epigenetic analysis and using methylation, DNA methylation, because DNA is better preserved in FFPE tissues than RNA, particularly if you go into very small samples. This is a picture I showed you before. So what if you have a tumor that you know has the presence of these amplifications, either in the same cell or in different cells, and how do you identify them and how do you profile them? So this is one of the tumors that we have profiled, and you can see this is really extreme level of heterogeneity. This is one GBM that has amplifications of NMYC, PDGFR-alpha, EGFR, MET, and CDK4, and there's absolutely no way to know which of these amplifications are mutually exclusive, as I've shown you in the previous slides, and which of them are present in the same tumor. So how do you go about it? So thinking back about the studies that we've done showing that MET protein is expressed or overexpressed in cancer cells that have MET amplification, you can see here an example of it from the GBM where we stained for CDK4 and EGFR. We knew that both of these genes were amplified in the cancer, but we didn't know whether they are co-occurring or mutually exclusive. You can see here in green cells are strongly positive for EGFR. There's only a little bit of a weak blush for CDK4. And here you can see with an overlap. So this is an example where you can identify purely EGFR overexpressing population, pull it out as a small pool, and then do downstream analysis on a molecular level. When you look in this, it's from the same tumor. You can see here the situation is completely different. This is staining for CDK4 and PDGFR alpha. You can see that cells here are strongly expressing both CDK4 as well as PDGFR alpha. You can see it nicely here in overlapping channels. So these cells very likely co-express the gene. You can then take this pool of cells and confirm in a part of the sample that you indeed have co-amplification happening in this subpopulation, and then work with these cells separately, and you know that this cell population has these two genes amplified. We are also pretty successful looking into antibodies for phosphorylated sites on the receptor tyrosine kinases. That's quite important because we are working with FFPE tissue, and from the perspective of functional studies, there's really not much you can do with dead cells, but looking into whether these genes are indeed functional or not, you can see this is an example of a tumor where we identified phosphorylated EGFR and phosphorylated PDGFR in the same cells. So that's also important. Phosphorylated antibodies are relatively difficult to optimize on FFPE tissue, but not impossible. So it's definitely something that can be done with a proper workflow. Downstream analysis that we use is DNA methylation. We use Illumina 850K DEPArray. There are other ways you can profile as well, but this is one that has been working for us relatively well. If you're asking why are we looking into methylation, it is easier to look at DNA methylation than on RNA expression, particularly if you're looking on a very small pools of cells from FFPE material, because RNA is can be extremely degraded, particularly if blocks of FFPE are relatively old. So here you can see, this is just how the subclones that we identified. Nicely, there's an epigenetic heterogeneity within the tumor. In strong contrast to mutational analysis, where you can really sequence deeper and deeper to identify mutations of your interest, epigenetics or methylation would be very badly affected if you have a mixture of cells. So if you have a GBM where there's a mixture of cancer cells, tumor-associated macrophages, endothelial cells, all these cells have completely different methylomes that, of course, have completely different RNA expression profiles, and not being able to dissect them will completely skew your results if you're looking at any kind of epigenetic regulation of cells. And of course, if you have a tumor that is driven by a cell that is driven by EGFR signaling or by MDM2 signaling, if those cells are going to be different, it's going to again skew your epigenetic analysis. So this is really sort of the summary of the chapter 1, and it's really how to explore the power of antibodies to detect aberrant protein overexpression in DEPArray. You can work with RTK antibodies like EGFR MET and PDGFR alpha, as I showed you, but you can also use, for example, ALK, which is overexpression strongly associated with ALK rearrangement in multiple cancers, or for example, HER2 if you're interested in HER2 amplified cells. Of course, I would urge you always to first optimize your thresholds to make sure that the cells that you're picking up with a protein are indeed the cells that have amplification of the gene or rearrangement of the gene. And as Raimo showed you, you can definitely confirm by FISH on cells that are isolated from with DEPArray. We're going to switch now gears a little bit into a different story, into how do you isolate the cells that you think that are relatively homogeneous genetically, but they're infiltrating through a lot of normal tissue, and you really need to pick up just the tumor cells. So this is just a little bit of an overview for those of you who are not familiar with the field of low-grade gliomas. These arise mostly due to early IDH mutations, and then even obtain 1p19q co-deletion and develop into oligodendrogliomas, or they do not have 1p19q deletion, but they have mutations in p53 and 8TRX, and then develop in astrocytomas. So all these can be used as markers for your analysis. So this is an example of IDH mutant astrocytoma. Here you can see H&E picture, very densely cellular tumor. It wouldn't be difficult to isolate these cells. You see the tumor cells have lost ATRX expression, which is present in endothelial cells. That's your internal control. It's also strongly positive for p53, which is not a mutation-specific antibody, but it can also be used. And then the most beautiful is the IDH1 R132H antibody, which is a mutation-specific antibody. So you know that every cell that expresses this protein, it indeed carries the R132H mutation. That has been shown extensively in multiple studies. So this is the problem that we are facing. We have a section of the brain on H&E, which is somewhat hypercellular. You can see here in the surface, there's more hypercellularity. You can tell by Ki67 proliferation marker that there are proliferating cells migrating through the brain. And when you do R132H immunohistochemistry, you can clearly observe positive cells in the background of a lot of negative cells. So now you can imagine if you did either RNA analysis or methylation analysis, you would have a mixture of mutant and normal cells. You would have normal glial cells. You would have neurons. All these cells have completely different transcriptome and methylome. So you really want to pick or select only IDH mutant cells to see what is happening with them. So this is example how you can do it. These are some of our cell panels pulled from DEPArray using the R132H IDH mutant-specific antibody. You can clearly see here on a DAPI, and then the red channel, you can see IDH, and this is the overlap. These are really beautiful IDH mutant cells. You can, of course, I would again urge you to optimize your workflow so you know what threshold you're having to pick the mutant cells. This is from the same tumor, IDH negative cells that can serve your control. You, of course, want to prove that what you selected as IDH mutant, it truly is IDH mutant and not the other way around. So you can see when you do methylation profiling, your IDH positive and your IDH negative cells would cluster completely separately, which again bodes strongly for that you're able very precisely select only cells that you are interested in using the specific mutation-specific antibody. And then you can do everything else with the cells, whatever your downstream application is. This brings us to a topic that we like to utilize in my laboratory, and that's really exploring the power of mutation-specific antibodies for DEPArray. Raimo has shown you that you can nicely select keratin-positive cancer cells and vimentin-positive cancer cells, but where I see the strength of this technology is we know what some of the founder mutations are, and we want to explore further how cancer has evolved past the founder mutations, and you can use this large array of and constantly growing array of antibodies, such as we are using, let's say, IDH, but also, for example, in lung cancer, EGFR L855R for the deletion, histone H3 mutation. These are a little bit different BAF47ATRX because they're loss of function. So you'll be looking actually for negative cells to identify your cancer cells, and then you process them with whatever your downstream analytics is. That brings me to the last chapter of today's webinar, and that's looking into a different question that we have recently started tackling, and that's heterogeneity of a brain tumor microenvironment. Particular macrophages, which are bone marrow-derived cells, part of a monocyte macrophage system, and in brain tumors, they're particularly cleaners of the dirty stuff, necrosis, hemorrhage, but they play critical functions in angiogenesis, inflammation, and immune response. And when you look just in CD163 immunohistochemical stain, you can clearly see that by morphology, macrophages in the viable tumor are very different than macrophages in the necrotic. These are large, they are foamy, they really accumulate the necrosis. They really just clean necrosis. But these guys are very interesting because we think they play a critical role in supporting tumor growth. And you want to be able to pull them out and see what's going on with them. TAMs play many important roles. They can be roughly subdivided in M1 and M2. I don't want to spend too much time on this. These are very well-known data. And we have shown that tumor-associated macrophages increase significantly after antiogenic therapy, and it's associated with a poor survival. You can see here how the number of tumor-associated macrophages goes up after treatment with cediranib. We have also shown later on that there is a marked increase after anti-angiogenic therapy, again associated with a poor outcome. But the real question is, what is the TAM population in GBM before any therapy, and what does it do? When you look at GBMs that are both the same, you ask yourself a question, what is the TAM population in there? And the answer is, it's very variable. These are two tumors, let's say they would be driven both by EGFR, and you can see in this glioma, there's a huge number of CD163 positive cells, and in this tumor, also malignant tumor, there are very few CD163 positive cells. And it begs the question, what is the difference between these gliomas, and what is the difference between their tumor-associated macrophage populations? So how do you get them out? So again, using DEPArray validated with using CD163 stain, you can clearly show that you can pull out cells that are positive for CD163, and there's only a little bit of a blush for EGFR. When you overlap the images, you can clearly see the cells are red. They express CD163, they don't express EGFR, and if you do your downstream analysis, you would, of course, you can also use the ploidy factor that Ryan was talking about. You would show that these cells are diploid as opposed to GBMs. And the best thing about it is that you can also identify things that you did not know about, and you wouldn't even think about the questions to ask them. So this was the project that we purely just wanted to pull out macrophages from different gliomas. But we have also found that in some of the tumors, we can actually identify a very small subset of macrophages that express not only CD163, but they also express EGFR. And that was a big surprise to us. There's a small subpopulation of macrophages within the macrophage world in the GBM. This has been, I think, shown before in breast cancer, that some of them can overexpress EGFR. So you can dissect heterogeneity not only at the level of cancer cells, but you can dissect heterogeneity on the level of a tumor stromal cells. And you can extrapolate that beyond just brain cancer. You can look into other cancers as well. You can look into other cells like endothelial cells, inflammatory cells, and so on. So you can see, you can have your scientific questions, and then you can identify subpopulations of cells that you didn't even know exist in the cancer that you're interested in. So in conclusions, I would conclude that tumors show genetic, epigenetic, as well as microenvironmental heterogeneity. That heterogeneity contributes to the therapeutic resistance and progression. You can dissect heterogeneity on a single cell level from archival FFPE tissue using dielectrophoretic capture digital sorting, which really allows you to handpick cells that you want to work with and that you're interested in based on your hypotheses. You can capture these specific subpopulations of cancer cells, but also tumor stromal cells. And then you can do downstream analysis that is interesting to your specific questions, be it a DNA analysis, RNA analysis, or analysis of epigenetics, as we are doing. And of course, this work was strongly supported with technical support and everything possible with Silicon Biosystems. Our founders, the Friedberg Charitable Foundation, Solon Conference Foundation, Making Headway Foundation, which support our brain cancer profiling efforts with people in our department and histology core and Department of Neurosurgery. Thank you for your attention, and we are happy to take any questions.
Thank you, Mattia. And so we have received several questions. I will start with the first one is how does FFPE processing affect the methylome of cells compared to fresh cells? And how many cells do you need for your analysis? I think this is for Mattia.
So that's a very good question. So the group in Heidelberg, German Cancer Institute, they have done extensive comparison between methylome of the fresh frozen sample and FFPE samples, and they've shown that there's a very good correlation. So you don't really, they actually correspond to each other very well. So you can really take the FFPE samples and extrapolate them to the fresh frozen samples. So I think that, you know, while DNA from FFPE samples is not as good of a quality, the methylation data you're going to be getting are equal to fresh frozen. Illumina has a DNA restore kit, which is used for FFPE sample, which helps you increase the quality of the calls from the profiling.
Okay, thank you. Then I have another question, which is what are the most common problems with preparation of brain tumor samples from FFPE for DEPArray?
I think the most common problem would be if samples are very necrotic. Necrosis can really influence the quality of your preparation because you will not be getting pure cells. You're going to be getting a lot of necrotic debris. So it's very important to carefully inspect the blocks and trying to find really viable tumor.
Okay. Then this question for Raimo, does this process work with regular medium?
Regular medium, we normally use, if I get the question right, and maybe Nicolò, you can also help me, we use two kinds of medium, one for fixed cells and one for fresh cells. So if regular medium is a medium that you use to keep your cells, to grow your cells, then yes, it's possible to use a medium for live cells. This is only causing us a bit of a slower motion of the cells inside the chip, but other than that, it can be used.
Okay, and then we have another question for Mattia. How well do immune histochemical stains used in clinical practice work for DEPArray, and whether you can use the same stains that are routinely used in clinical practice?
Yeah, so we try to actually always start with the antibodies that we use in the clinic because we know those are really robust. So we always start with those antibodies and we try to optimize them for IF. It's not always possible, and then you have to shop around for different antibodies, but we've been pretty successful. It always takes a little bit of tweaking to get the antibody working. The good thing is that DEPArray has extremely sensitive camera, so even if your signal on the regular fluorescent microscope is relatively weak, once you load the sample into DEPArray chip, it's actually much brighter than what you thought in a fluorescent microscope. So you have a much better time to select your cells, even if the signal seems sort of like weak under the standard fluorescent scope.
Okay. And then we have another question. Beyond keratin and vimentin, do you suggest other markers for selecting stromal and tumor cells? In particular for neuroblastoma, as vimentin positive may exist, do you suggest other markers for stromal cells?
So I work also with brain tumor, so we don't actually use much of vimentin. When you look at the stroma, it really depends what stromal population you're interested in. I think you can use SMA for fibroblasts. That's one of my favorite markers. And then we really focus a lot on endothelial cells and on tumor-associated macrophages. And then it really depends what kind of other population you'd be interested in. I would really urge always people to go for a specific. Vimentin is not the most specific antibody. If you're looking for specific population, you really want to dive a little bit deeper and have as specific marker as possible.
Then I have another question for Raimo, whether there's a capture bias in the DEPArray energy minimum points due to cell size or shape?
Well, the system is capable of capturing and moving in a same experiment cells that range between, say, 5 microns and 55 microns. So just to give you an example, in the forensic area, we work in the same experiment with sperm, blood, and epithelial cells. Larger than that limit, the cells are becoming difficult to move, and smaller than that limit, the cells tend to not be capturable by the cage, so they will be not, let's say, manageable by the system. But within this range, I would say every cell can be managed easily.
And there is another one for Mattia, and it's about which enzymes do you use for disaggregation of brain? How do you disaggregate the FFE?
Yeah, so you really have to tweak your enzymatic cocktail based on how the tissue looks like. We started with a standard cocktail provided, with suggestions provided by Silicon Biosystems, but we also experimented a lot with acutase, papain, and hyaluronidase. In my experience, what is really critical is to sort of like look at the tissue and see how densely gliotic it is, or how loose it is, and mix it, how rich it is in glycosaminoglycans. If the tissue is very rich in glycosaminoglycans, adding hyaluronidase really helps. Collagenase is also good to start with, but brain doesn't have a lot of collagen, so being too heavy on collagenase doesn't necessarily help you. Trypsin, we had, I think, 0 success with. Some enzymes could be great in making suspension, but they can be dangerous to your epitopes. So if you incubate for too long or you have a too high concentration of enzymes, you can kill off your epitopes. So it takes some tweaking and really understanding your tissue and try a few iterations with markers that are supposed to work and that you have them. So what we always do is we always first test the marker by immunofluorescence on a regular FFPE 5-micron slide. We get it working by IF, and then we start working on the suspension. And so to make sure that the antibody is first working, and then you have to mix your enzymes in a way that will help you achieve the good suspension without killing the epitopes.
Okay, thank you. And we also have another question which came during Raimo's presentation, which is what is the voltage and RF frequency used by DEPArray, and is that stressful to the cells? Now, Raimo, I know this.
You can answer for me, Nicola, please.
I can help you because this is very technical, I understand. So we use megahertz frequencies in the chip with low voltages. So using megahertz frequencies with just probably, I think it's 1.5 or up to 2 volts peak to peak, there is no transmembrane potential induced when you use live cells, so the cells are not damaged. And okay, and I think that with this we are basically in time for the closure, just in time. And I would like to thank again both our speakers and Elizabeth. I don't know if you want to add something more.
Thank you so much to all of you.
Thank you, everybody. It's a pleasure.
Thank you, everybody.
Thank you. Bye.